The cell biology of infection, or cellular microbiology, it's really synonymous with advances in microscopy techniques. New microscopes are evolving faster than iPhones. It's really an amazing time to visualize the infection process at a resolution that was just previously not possible. And in order to visualize infection from the level of the single cell to the whole animal, we use the zebrafish larva. So in terms of challenges, it's really to be able to follow the cells or the infection process at a resolution that's not possible to achieve in other labs. And so you need the highest end resolution microscopes, but you also need better tools to analyze and to understand the infection process. But it is an exciting time because tools such as the Athena software are enabling us to quantify these things, I guess, at a pace and at a resolution that was not previously achievable.
My project is focusing on investigating the evolution of enteropathogens. And to do this, we essentially collect a bunch of strains of different backgrounds and we infect zebrafish with them to try and understand what makes one strain better than the other. When we're looking at the zebrafish, what we're looking for is differences in host response, so how the number of neutrophils and macrophages in a zebrafish changes over time. And this will help us understand why some strains are able to infect humans better than others.
We're able to move away from the infection of, say, half dozen, a dozen larvae at a time, to now we're starting to work with 96 well plates, because now you can image things at a much faster pace using these new microscopy techniques. However, with these different microscopes, you really need the innovation of analytical tools. And that's where Athena could really help us, because although we were generating data at a much faster pace, it was no longer feasible for students to kind of manually quantify all these things. It simply takes up too much time.
Before using Athena, I would actually typically count cells by hand. And zebrafish can have up to 200-300 cells, so doing this for 60 different fish at a time was really challenging, and it would often take me hours to sit there and count manually. So using Athena has massively accelerated the progress that I'm able to make.
And so in this way, we can now just collect data over 2-3 days, because all the analysis is live, at least in our case with Athena. And then we just plug these images into the Athena software, and literally with a few clicks of the button, we can now analyze our areas of interest in the zebrafish larva and reliably quantify things at a scale that was previously unimaginable.
I've been able to include more strains that perhaps I wouldn't have been able to include before, so we're able to get a broader picture of how the background of these strains look as compared to using just a couple of strains. So it's improved my science in that way, and of course it's allowed me to have more time. Since I put the images into Athena, it automatically does it all for me. I can walk away and do other stuff. And I really like the features of being able to kind of set the parameters once and then just going back to it and using that same kind of experiment for every single one, so you don't have to define the parameters each time. Once you've done it once, you're kind of set. That's something that's really stood out to me and has changed things for me. I have a direct line of contact and they're super helpful. So I'm able to share my screen with them and they will log on and see what problems I'm facing, or I can send them a log file for the computer and they'll be able to deduce from that what the problems are. And they're really good at getting back to us really quickly, and they're typically resolving it the same day.
It was just a great pleasure to work with the Athena team in order to kind of test the systems they have in place and to give them ideas on how to push it further. So that's one way. We would like to push the softwares. We would like to help innovate the softwares that could really benefit the zebrafish community as a whole. As we publish our research, as we help to develop these softwares, I think it'll make for a more robust science across the entire community.